Well, good afternoon, everyone, and welcome to this webinar being hosted by the U.S. Forest Service and the Association of Fish and Wildlife Agencies. My name is Dr. Jonathan Maudsley. I am the Science Advisor at the Association of Fish and Wildlife Agencies. We're delighted to be able to co-present this webinar today entitled Assessing Surveying Methodologies to Assess Information Gaps for Forest Carnivores in the Pacific States with Dr. Katie Moriarty, Research Wildlife Biologist at U.S. Forest Service R&D Pacific Northwest Research Station. A couple housekeeping things before we get started today. Once we turn it over to Katie, I will be muting all the lines so that we won't hear any extraneous noise, people typing, etc. during the webinar, and we do appreciate your participation today, and we're glad to have so many people with us. We're getting a great turnout, I can see here on the control screen. We will be muting the webinar. We're also recording this webinar, and we do plan to make these available through the AFWA website at some point in the near future. If you would like a copy of the recording, please feel free to reach out to myself or John Roethlisberger at U.S. Forest Service. We will be also taking questions. If you have a question for the speaker, please use the chat box in the lower left-hand corner of your screen. You're welcome to send a question. I will receive that here in the control center in AFWA, and we will be reading those at the end as time permits and giving Katie an opportunity to respond. We do try to keep these webinars to an hour in length. If we do finish early, we will have time for audio questions and answers, at which time I will unmute the line and have opportunities for dialogue with the speaker. But we do appreciate all of you tuning in today for this webinar, and we appreciate the opportunity to co-sponsor it with our colleagues at the Forest Service. And here to say a little bit more about the Forest Service and to introduce today's speaker is my good friend and colleague John Roethlisberger from the Forest Service. John.
Thanks, Jonathan, and good afternoon or good morning to everyone who is out there. This is John Roethlisberger. I work for the U.S. Forest Service Research and Development staff in our national office that is tasked with fish and wildlife research. And the Forest Service appreciates this opportunity to partner with AFWA on this webinar series. Our goal in this series is to share the latest scientific research and synthesis that is relevant to fish and wildlife conservation and management. And I think we've got a great opportunity to do that today as we hear from our speaker, Dr. Katie Moriarty. Dr. Moriarty is a postdoctoral research scientist with the U.S. Forest Service Pacific Northwest Research Station. She is based in Olympia, Washington. She graduated from Oregon State University with a PhD in wildlife science in 2014. Her dissertation research for that degree focused on the effects of forest management practices on habitat use and movement behavior of Pacific marten. Dr. Moriarty has continued in that research theme, and her work focuses on the distribution, demographics, and status of small forest carnivores in Oregon, Washington, and California, with a focus on the implications of these movement patterns and distribution and demographics for forest management. So now we are very fortunate to have the chance to hear from her, and I'll turn the time over to Dr. Moriarty.
Thank you. Thank you.
Great. I'm going to mute all of the lines and bring Katie back up. Thank you, Katie.
The conference has been muted.
Okay. Katie. You're on.
Okay. Thank you. Today I'll be talking about carnivore surveys, detectability, and monitoring. John, can I bring up my PowerPoint slide? Perfect. The accumulation of this talk is going to involve a great many projects, and I'm listing a lot of my collaborators here, but this is not an inclusive list. One of the primary themes for this talk focuses on how much is possible with collaborations, and I'm really excited to highlight our recent work in the Pacific Northwest, which was only possible with a team of university, federal, state, and private agencies. My goal, by and large, is to provide just enough detail for a diverse audience to learn about these species and survey methods. But my hope really is that the talk provides enough information to fuel questions and conversations among your peers and respective agencies. Certainly this talk is carnivore focused, but I feel that the themes will apply to a diversity of species and communities. So hopefully everyone can get something from this information. At the end, I'm introducing a monitoring vision, and I want to make it perfectly clear that we're in a very early phase of planning. Such a vision will only succeed if we have the ideas, support, and continued collaboration of a lot of different landowners and scientists, and it's not, or at this point, it's very inclusive. So with that, I'd like to begin. Next slide. Perfect. So to give you a roadmap, first I'll go over some background, and then I'm going to use some of the work in the Pacific Northwest as a case study to basically show what we can do with distribution. And then lastly, I'll caution that such distribution surveys really rely on detectability and the ability to use survey methods that are going to give you reliable estimates of whether or not these animals are present. And we're at a place where we would really benefit from a cohesive and purposeful framework that is fairly consistent across a large geographic area. Next slide. If you think about carnivores broadly, there's been several examples, especially recent examples, on how some of the larger carnivores can very much increase biodiversity and ecosystem resilience. Next. And even some of the mid-sized carnivores for which we have enough data vastly can affect their ecosystem. Next. Such that you can visually see differences in areas in which, in this case, sea otters reside and where they are no longer present. Next slide. But besides from ecosystem impacts, there are other reasons to be knowledgeable about these species. For instance, in the western states, we have a variety of political designations that we may be inclined to be aware of. Lynx are the only federally threatened species. Next slide. However, wolverine, fisher, marten, and red fox, specifically montane red fox, have been petitioned for listing. Next slide. 3 of those species have been warranted but precluded. Next slide. And the listing decision has recently been withdrawn for wolverine, fisher, and marten, and for each of those species, litigation is currently in progress. Next slide. At a state level, 4 of these species are either threatened or endangered in Washington, Oregon, and California. Martens are conspicuously not. In coastal California, they are being considered as a state threatened species, and in Oregon and Washington, they're still trapped. Next slide. As the Forest Service, we are land stewards of a very large area in the western states, and we have the intention of providing for the persistence of all native species, contributing to the recovery of threatened and endangered species, conserving proposed and candidate species, and maintaining species of conservation concern. And to do this, I would argue that managers need both information and tools. Next slide. So as an example, I'd like to showcase martens because, at least in this case, they're the smallest of these 5 carnivores. They are the most abundant, at least in the western states. They have very similar life history characteristics to wolverine and fisher. And likely we can use them as a test case or a canary in the coal mine to start to gauge some of our management information. Next slide. Martens are generally considered, at least in many places, a boreal species. Next slide. And they're often, if we do a quick comparison to, say, the endangered northern spotted owl, they're very similar in size and most aspects, except for their critical reproductive period is longer and their survival and life expectancy is shorter. Next slide. Martens are considered a habitat specialist. They're a cavity denning obligate. They use resting sites or locations that have cavities or platforms many times a day, many different structures. These provide refugia from predators, temperature extremes, and precipitation. And at least in California, these structures are extremely large and very old. Next slide. Martens, however, are a dietary generalist, which means they'll eat anything. They have a diverse amount of prey available to them in the summer, but in the winter they're likely limited to non-hibernating mammals. And food is really important. Next slide. Martens eat about a quarter of their diet or body weight every day. So for me, I have no intention of eating 100 hamburgers today, but if you can do that, that would give you an idea of how much a marten is trying to eat on a daily basis. Next slide. And their movement is really important. So that prey fuels what we're learning, a really intense amount of activity. Next. In fact, some of our GPS collar research suggests that they can move almost 2 miles in an hour and long distances 11 to 27 kilometers in a day. And if you compare the weight of a marten, it's very similar to a fox squirrel or a gray squirrel, about a third the size of a house cat. So these animals are moving extensively and in broad areas. Next slide. On a daily basis, both males and females in summer and winter are moving about 6 to 8 kilometers on average every single day. So this particular slide is the average and 95% confidence interval. Next slide. Their movement is really important because it's been shown that they're more likely to encounter, attack, and kill prey in unmanaged stands compared to previously managed or regenerating areas. Next slide. And in addition, next slide. Okay. In addition, we've also done experiments. Oh, one back. John?
Yep.
Okay. One before, please. Okay, now one forward. Perfect. In addition, we've done experiments that have revealed that the odds of detecting marten in the summer are over 1,000 times less likely to adjacent unmanaged stands. And similarly, they're 100 times less likely to be detected in thinned areas for fuel and fire reduction. And this is because of the experimental nature of this particular aspect and study, likely due to predation. And if you recall by the size, anything that can eat a squirrel can also eat a marten. Next slide. And at a population level, it's been shown, at least in 3 instances at a landscape scale, that as soon as you move as little as 30% of the forest cover, the entire population could become locally extirpated. Next slide. So I used martens as an example. Certainly this is probably not unique among the carnivores. They all have life history characteristics that are really reliant on both their prey, their structure, vegetation structure, and potential predation. And I'd like you to keep the various aspects of these life histories in mind as I continue forward. Next slide. So one of the foundations in management is to be able to understand distribution of the species, and this certainly has been necessary in the Pacific Northwest. To give you an example, next slide. In coastal Oregon in 2014, with an impending listing decision for coastal martens, Oregon had a total of 26 locations of known marten, of which 14 were roadkill and 3 were legally trapped. Next slide. During a large multi-stakeholder meeting, it was determined that the knowledge was insufficient for developing management alternatives or directions. Next slide. And luckily, powerfully, a lot of collaborators joined in a very short time period to try to understand minimally where these animals were and if the populations were still persisting. This involved not only a lot of funding agencies, whose logos are shown here, but also numerous landowners from across the gambit of all of the places in Oregon. Next slide. Within less than a year, we were able to survey a majority of the prior range of the species, and it was very, very difficult work, as you can see from some of these technicians' traveling experience. The Coast Range of Oregon is not to be lightly discussed. Next slide. And this particular effort was completed by the help of 6 field teams and 5 different agencies, surveying on 10 different ownerships of land. And we did get donations, small or large, from 10 different agencies. Because this particular event was so successful in a short period of time, in less than a year, with almost 1,000 camera stations over 428 unique areas, we were able to double this effort. Next slide. So less than a year later, we increased not only for marten in the Coast Range, but for fisher in the southern Cascades and Klamath Mountains, a little bit more surveys in the north and also on the Olympic Peninsula with a variety of stakeholders. Within another year, we had more funding agencies, increased collaborations, and 2,000 camera stations that are running for over 2 months is a non-trivial effort. At this point, we have about 4 million photographs, not only of our target species, next slide, but also potential predators, next slide, and potential prey. Next slide. In terms of tangible results, we did find martens in about 56 locations in the Coast Range. Since then, this has increased with also additional survey effort. We were able to obtain about 300,000 photos overall and 100 scats from this effort alone. These data are being entered into what's called the NRIS for the Forest Service Wildlife System, or GeoBob, which means that the public landowners will be able to use these data and integrate them into their management plans. Next slide. Similarly for fisher, this work is in progress with a master's student, Brent Berry. We're trying to understand the distribution and detectability of these species, and we have some interesting preliminary results. Next slide. At this point, it seems like the distribution is much less than was priorly depicted, and we're hoping to use our data to help identify areas to restore or place additional research in the future. Next slide. But this is only a snapshot in time. We've expanded our surveys, so now in green we have surveys that we're going to do next year. All of the depictions in gray have already been completed, but it's not necessarily monitoring. It's more of a snapshot. Next slide. And to set up a monitoring program, we really need to consider how detectable these animals are, and it takes a lot of different nuanced factors into mind, like the protocol itself, the season, the baits, the units, the spacing, and we're trying to move forward to address some of these issues. Next slide. If we look at trying to develop a statistically robust framework, there has been a recent spatially explicit power analysis that has been published for wolverine. We expect for most of these carnivores, we have about a 20% detection probability. Oh wow, and my computer just shut down. One moment. The hazards of not being able to control your own slides. Sorry. So we have about a 20% detection probability. Typically we visit camera station or track plate boxes, these non-invasive survey devices, on multiple occasions to replace their baits. Occasionally this is 2 times. It could be up to 7 in this particular simulation. Next slide. And if we look at our capacity to understand population level declines, it's not possible, even with 300 or 400 camera stations or cells sampled, with a small percentage of population change. Next slide. To be able to detect a 50% decline, it's nearly impossible with a 20% detection probability. And even if we get to an 80% detection probability, which are indicated by the dashed lines, a 50% decline in population is fairly significant. So this suggests that you need many cells surveyed, in this case over 100 in this simulation, up to 400 to have high confidence. And that effort is non-trivial. And this isn't just a simulation. Next slide. Empirically, we have evidence. One of the larger fisher studies, at least in the Pacific states, was through the Sierra Nevada Adaptive Management Project. This particular effort had over 100 radio collar fishers over a 7-year period. And luckily, they radio marked their antennas with reflective banding to be able to look at individual identification. So paired with their radio telemetry data, they also had about 1,000 cameras spaced 1 kilometer apart. Next slide. And with radio telemetry data, they were able to discern that if the animal was within 250 meters, which is not that far away, the probability of detecting animals on average was around 25% in the summer. And if there were 2 visits during the winter, that increased to about 60% probability. So these are fairly low probabilities to detect the species. Next slide. And even that difference between summer and winter can have huge implications on how we perceive and interpret what habitat and connectivity is. So for instance, with Marten data in a different location, using detection, non-detection data, the summer looks and appears to have much less habitat than the winter. And it depends on what your goal is to try to interpret how each of these should be represented and thought of. Next slide. So at least in Oregon, what we're doing currently is trying to understand if we can use methods to increase our probability of detection. We're building on survey methods that have been used in the Pacific Southwest Research Station of California for over a decade, where 3 cameras are placed about 1 kilometer apart in a cluster. And cluster designs, at least for some survey metrics like spatial capture recapture, have a higher capacity to provide more accurate estimates of precision. And in addition to these clustered baited cameras, we have a trail camera that is within that same unit to try to look at whether or not species are detected differentially based on bait and perhaps avoided or are avoiding baits. In addition, each of these baited stations has a different treatment that is randomly selected. So for instance, in the bottom left-hand corner, we have a station that has not only cat food and chicken and an apple and grains and a salted block for porcupine. And that contrasts with, say, a station that just has chicken or a station that just has cat food. And we're able to do these replicates on a fairly large scale. All of Oregon has, at least at some point in some season, some variation of this protocol. And that's a huge sample size to be able to look at differences in detection based on bait or camera set. Next slide. Preliminarily in the coast, we are getting information that shows there is a difference in how animals are perceiving these baited stations. For instance, a very expected result, but still nonetheless, is that felids by and large are detected more on trail stations that are non-baited than they have been in areas that have had any particular bait. Conversely, mustelids like martens and fishers are detected more frequently in areas that have bait and lure than a trail set. So that's just one example. But to get even more exciting, next slide, we're not only using camera stations, we're also using scent detection dog teams that have been extremely well trained, hundreds of thousands of hours on the species in particular interest, and each dog, at least for our study, has been trained to identify scats of 6 species: marten, fisher, red fox, porcupine, which is a potential prey species, and also mountain lion and bobcat, which are potential predators. So we're conducting these paired surveys where dogs are going to the same areas and vicinity as cameras, in addition to areas that we have unknown data. Next slide. Preliminarily, again in the Coast Range, with a fairly small sample size, the dogs are doing quite well. We were able to detect marten at 24 sample units with paired cameras and detection dog teams. The detection dogs that genetically verified through SCAT were able to find marten at 22 of those 24 stations, where cameras were able to detect marten at 12 of those 24 stations. So generally with our particular study, it was cheaper for the detection dog teams to look at distribution. However, it was more expensive to use this survey method if you were trying to understand individual ID and sex. It's a difference between mitochondrial and microsatellite DNA amplification, and the SCAT amplification for individual was only 17%, meaning that we needed to go and get more scats to be able to identify a diversity of individuals. We're working with collaborators at Oregon State University to use Illumina sequencing and next generation sequencing to see if we could increase our capacity to look at individual and sex, which may become important later. Next slide. In addition, we've basically doubled this survey effort where we're using detection dog teams in concert with the remote camera stations, in concert with GPS collared data from both fisher and marten. So here we're hoping to be able to assess not only which method is more efficient, but at what radius can we expect our sample units to have the most impact and reduce our capacity to have false absences. Next slide. So in progress, but I can't report on today, are surveys that are at a very broad scale, trying to look at various aspects that should affect detectability. And we have treatments to try to assess the different impacts of each of those various factors. Next slide. However, this doesn't really get at demographics or trends, which is something that I think everyone wants to know once you figure out where these animals are. Next slide. And that is really difficult because unlike animals that use, say, acoustics to move or to mate, or animals like bull trout that are restricted to streams or corridors where you can measure eDNA, carnivores roam widely, and we don't know whether or not it's beneficial for them to come to a baited station or an unbaited station. Next slide. And from trapping records, carnivore detections can often be dominated by juveniles and males, and we need to figure out how that relates to demographics and trends if we're to use non-invasive survey methods at a broad scale. Next slide. And this is really going to be important when we're trying to identify relative risks and what is not only at a spatial scale, where are we concerned with management activities, where do we need to potentially focus restoration efforts, and what could happen if there's unforeseen events such as fire. Next slide. So I'd like to propose with several collaborators a new monitoring vision, and I think that it's going to be really important to have a diverse network of scientists and managers to collaborate and develop a long-term collaborative, multi-scale, multi-objective, multi-species, hypothesis-based plan for cohesive monitoring. Currently this effort is led by the Rocky Mountain Research Station and Region 1. My colleagues Jesse Golding, Mike Schwartz, and Kevin McKelvey are basically developing the vision. Next slide. And it's really important for us to try to think about how we can work together to be most efficient. These animals are extremely wide-ranging. Currently, our systematic protocol that's a standard was created in 1995, and although it's used fairly regularly even today, next slide, there is a lot of new methods and new opportunities to be able to enhance those data and to increase our understanding while decreasing relative uncertainty. Next slide. The plan is threefold. The first is to analyze existing data. And we do so not only with the non-invasive survey methods, but also studies that are being in progress to try to assess effectiveness. Next slide. And for instance, we have a wealth of information already. In Region 5, there's been a systematic carnivore monitoring program that has occurred over the last 15 years. They have surveyed over 2,600 stations over a broad spatial extent, and this is unique in that they have repeated sampling over these units over time to evaluate trends not only in occupancy but distribution. There's a really powerful data set both in space and in time. Next slide. Recently, in addition to just surveying with non-detection data, they've increased to include genetic hair snares of various types on all sample units, and this allows them to answer increased questions not only on occupancy, but sex ratios, connectivity, minimum number of individuals, and isotopes will allow dietary analysis at a coarse scale. Next slide. If you combine that with the data that we're collecting, which is very similar and comparable in scale, and in scope and in number, we have the capacity to really understand what would be the most efficient monitoring program over an extremely vast area over time. Next slide. And this is going to be really important to start to develop a hypothesis-based framework that is very specific in area, spatially, and temporally, such that we can move forward and take our data and try to achieve and have a goal of a desired condition for, say, Fisher. And as an example, next slide, spatially in Region 1 and Region 4, we might identify areas in which we know that we have not done significant surveys to understand whether or not Fisher are present. So in the lighter green, Fisher distribution is relatively unknown, and we can temporally say that that would be our highest priority. Whereas the areas in a darker green also have a habitat suitability model, which is in orange, and we suspect that those areas are not only occupied but have higher densities perhaps. And we can use that as a null hypothesis to be able to build and understand the trends in those particular areas as a spatial example. Next slide. And as we find areas in which we understand and know where animals are, we can take those representative locations and start to build even further to assess limiting factors. So for instance, in the coast of Oregon, we have a master's student who has done a great job looking at small mammal indices of abundance and distribution by vegetation types. She's comparing not only the relative abundance with metabarcoding using Marten scats and diet to see where prey is located. And then simultaneously we have radio-collared individuals, a camera trap network that is looking for spatial capture, recapture, so we could estimate not only movement rates, use areas, but also minimum population size and overall density. Next slide. So lastly, as part of this framework, it's going to be important to not only build on our data, but also to continue to monitor and fill in all the gaps so we can decrease our uncertainty over time. And this is going to be particularly suited as a framework that's flexible to be able to address land managers' information needs over time. Next slide. So in summary, I highlighted 5 species in the western states that may be of concern or of interest, focusing primarily on marten and fisher. Next slide. All of these species are wide-ranging, at least in the western states, persist at low densities, and would be considered relatively rare. Next slide. I highlighted martens as a case study to try to explain some of the characteristics that carnivore has that are potentially a challenge for land managers, including a very restricted use of various vegetation structures that are most certainly related to not only resting refugia and denning, but also prey and trying not to get eaten by potential predators. Next slide. I highlighted essentially Oregon as a place where we have a numerous number of collaborations that have been able to work together through a very systematic network and survey a dramatic number of sample units in a short period of time. Next slide. However, such information is very difficult to get unless you have a cohesive monitoring plan. And ideally, at least in what I've found in the Pacific Northwest, it's going to require many devices and multiple methods to have reduced uncertainty on whether or not these species are present. Last slide. And I also proposed that we can work together to create a monitoring vision that's going to encapsulate a lot of collaborative ideas, but also move us forward so we have a cohesive and efficient monitoring plan. Next slide. Thank you.
Great. Thank you very much, Katie. I'm about to unmute the lines. We do have 72 people on the call at the moment, so there may be a little bit of background noise.
The conference has been unmuted.
We do have several questions that have been submitted here within the dialog box, and so we're going to go through those first. We ask for folks on the line to please either mute your phone or try to eliminate background noise. We are getting some background noise on the call, so please do mute your lines if you're not already doing so. Question here from Sean Murphy. This goes back to the work that you did, Katie, your field work to look at the cluster sampling. Did you use simulation and previously published parameter estimates, for example, DEP, et cetera, to establish the cluster sampling? And if so, what spacing between clusters and what parameter estimates were used for simulation scenarios? So a question there about how you established the cluster sampling.
Yeah, so we revisited work by, let's say, Catherine Sun has a 2014 paper in PLOS One. Clay Wilton also, I think, has a 2014 paper also in PLOS One. Those are specific to spatial capture recapture and reducing uncertainty and having increased parameter estimates. But ultimately, what we decided was to use the same protocol that's been done in Region 5 over the last 15 years and try to use different spacing dependent on the species of interest. So the spacing for marten clusters is a little bit smaller, I think 750 meters apart, versus for fisher, they were 1 kilometer apart. And by and large, I think the clusters themselves were half of a home range size for a female. But we also are doing surveys to test all of those various spacing, and we're going to use those data to try to formulate what would be the most efficient in the future.
Great, thank you. We have a question here from Diane McFarland, wondering at the lack of survey regarding fisher and marten in the Olympic fisher reintroduction area. Is that exclusive to National Park Service?
So I didn't show the Park Service surveys. Over the past few years, they have been surveying in very intensive detail to monitor their reintroduction of fishers. Those surveys were specific to look for marten. And in the Olympic Peninsula, there have been, thanks to our surveys, a total of 6 verified marten detections since the 1980s, 3 in the past 2 years. So that's another story that probably could be told all in and of itself. But there have been extensive carnivore surveys on the Olympic Peninsula.
Great, thank you. I see Diane has her hand raised. Diane, do you have a follow-up to that?
Oh, sorry, no, I just wasn't sure whether I needed to do that.
Oh, okay, very good, very good. Yeah, no problem, we got it, thank you. Question from Sophie Gilbert. What are your preferred camera and bait types?
Well, this is not a Forest Service opinion, but my personal opinion. Currently we're using the recent version of Bushnell aggressors. Those particular cameras have been in the field almost full-time for over 1.5 years, and we're getting some attrition, but not too badly. I would prefer reconics if we could afford them, but I think sample size is more important in this particular case. And for Marten, we designed a study to look at differences in height, low sets being within less than 0.5 meters to the ground, and high sets being at diameter at base height about 1.3 meters. And there's some evidence to suggest that lower sets are a little more efficient. So we're analyzing those data now. If your goal is to survey for marten or fisher, and probably montane fox. And we don't have lynx or wolverine in those study areas.
Great. And Sophie had a follow-up question. She says she'll follow up separately with you about the potential for expanding the collaborator network to Idaho and or Alaska. So,
perfect.
Yeah, a question from Katie Crumley. What types of marten and fisher interactions, factors such as habitat use, exclusion, trends in elevation, snow depth, have you found in the camera data?
That's a great question. We will be analyzing those data, at least for the cascades where they overlap. And in our study area in Lhasa National Forest, fishers were reintroduced recently. So we will have the evidence of fishers expanding and martens interacting novelly to those new competitors. I think John Linnell has published information in Canada showing that martens are avoiding areas with fisher. And our data are not analyzed yet to be able to reveal such patterns. But I strongly suspect there's going to be competition-level effects between the carnivores, not only marten and fisher, but of course bobcat and fox and others.
Great, thank you very much. Question from Bruce Marcotte. What do you envision for using decision science in crafting management guidelines from this research?
I think that's a great question, Bruce. I'm hoping to get your input on that. But roughly, I feel like we're going to have a framework that's going to be both spatially and temporally developed with a lot of uncertainties based on region. And I think structured decision-making can provide an opportunity for us to articulate that framework in a repeatable fashion, not only for these particular carnivores, but for other species. And that way our decision framework, similar to, oh gosh, it's a 2012 book that Bruce Marcotte is a co-author on. Either way, structured decision-making allows us to have a formal framework to be able to move forward and make decisions.
Great, thank you. That's all the questions we received in the chat box for the moment. We do have a little bit of time left in our scheduled 1 hour for this webinar. So if there are other questions from folks on the line, I would invite you to come forward at this time. Please state your name, your affiliation, and then your question for the speaker. So I would invite questions from folks on the line. Could you speak into the phone a little bit? We're having trouble hearing the question. Thank you.
Sorry, I'll try talking louder. My headset doesn't work well. So I'm wondering, Katie, what collaborative or additional data will you be collecting to look at questions like how the areas where these species occur are differing in their snowpack and the moisture content, et cetera, to address the climate change effect?
So I'm going to repeat some of that because it was a little difficult to hear. I think you were asking how these data can be used to assess potential impacts of climate change given martens being a boreal species and declining snowpacks. Is that true?
Well, what data will you be collecting for collaborative analysis?
Uh-huh. Well, there's some exciting things happening. First, just for the audience, there have been some various models that show, at least in California, which is the southern extent of the Marten Range, that snowpack is, or marten habitat more importantly, is expected to decline by 40 to 80%. And I don't have those numbers exactly, but it's significant. That's Spencer et al., 2015. And in our Lassen study area where we have radio telemetry data, GPS data, 8 years of marten movement data, we also recently have started a collaboration with Dr. Jonathan Pauly at University of Wisconsin-Madison to set out temperature logger data and try to look at marten energetics, we hope, in the future. And that should provide some information on how we would expect things to change given scenarios. But that work also is in progress and not entirely all the way funded. I think. What? You don't need any more.
Great, thank you. Thank you very much. I see Robert Long has his hand raised. Robert, do you have a question?
Yeah, can you hear me okay?
Yes.
Okay, great. Katie, great job. Amazing amount of information. I just wanted to clarify, early on in the presentation, you mentioned that you had found previously that the odds of detecting martens were 1,000 times less in open stands. And did you mean detectability and not occupancy? So even when they're present, they were 1,000 times less detectable?
Yes. So with that, I think, it's the odds of detection probability, but that particular experiment was conducted in areas where there were only radio-collared home ranges. So we know the martens were present during the time of the study, and we have them detected within their home range within the particular experiment at the same time that they were avoiding the openings and descent areas.
I see. Okay. Okay, that's great. Helpful. Thanks.
Mm-hmm.
Great. Any other questions for Katie? Great. Well, thank you again, Katie, for this wonderful presentation and for sharing it with our audience this morning. We had a great participation, 72 people engaged in this webinar presentation, so thank you again for sharing this with us. We are just absolutely delighted to be able to present this to the community of the state fish and wildlife agencies through the Association of Fish and Wildlife Agencies, and to our colleagues in the Forest Service through the good work being done by Katie and others and John at Forest Service R&D. And so for some last words, I'd like to turn it over to my colleague, John Roethlisberger from the U.S. Forest Service. John.
Okay, great. So on behalf of Forest Service R&D, I again would like to thank Katie for her excellent presentation today, and also say that we appreciate everyone's participation in the webinar. Thank you for tuning in. Thanks for the great questions and comments, and hopefully there will be future opportunities for collaboration that will grow out of the information that Katie shared today. Please feel free to reach out to her or to any other Forest Service researcher for assistance or with opportunities to collaborate. Jonathan and I can help make connections as needed also. I'd like to say thank you to Jonathan Maudsley and to the Association of Fish and Wildlife Agencies for organizing and hosting this webinar series. The next webinar in this series is scheduled for just about 1 month from now. That will be Tuesday, December 13th, and that presentation will feature Dr. Todd Wilson of the Forest Service, who will be speaking about forest stand structure and small mammal biodiversity. We greatly appreciate the feedback that we receive about these webinars, so when you receive the feedback form after the webinar, please fill it out. And we are definitely interested in suggestions for future topics as we continue the series. So please share those ideas. Thanks again for everyone participating today, and have a great afternoon.
Yep. Thank you very much. This concludes today's webinar. Thank you.